Thanks, Kathy. Thanks, everybody, for hanging with us here throughout the day and a half. I always reset when I come here, and so looking at art, it helps me go back home and dissect between the urgent and the significant. And boy, do I have problems, you know, the tyranny of the urgent, right? But can you play defense against yourself and all the surroundings of daily life to focus on the significant? And I think this project has lots of elements of that, and I'll give you a guided tour through proteomics and protein-based analyses. And that title has a pretty big claim, complete molecular specificity. So it is possible now, and I'll show you how that's done, to define protein molecules intact or even in whole complexes and characterize every atom properly. So this is in the context of modern omics research, and so I'll drift through there in the first 4 minutes and talk about mapping cell types and using proteins. What are the rules about how we would define cell types using proteomics technologies of 3 flavors that we'll get into? And this is in the context of Bcell lineage mapping. So in primary populations of human, in blood cells and in bone marrow cells, how can we use these, define the rules of proteomics to define Bcell lineage, how much can be done, and what are the 3 flavors of proteomics, how do they contribute to this process? And then at the end, I'll just give you a few minutes about the prospects for how we can define the baseline of knowledge in cells and the proteins within them to advance basic biology and translational research. And unlike the floor has these DNA molecules on it, we have a really firm basis, right? But in proteomics, there's some open-ended questions. Can we close those off this decade? All right, so if we take DNA, the genome project, I'm just going to give you 3 overlapping Venn diagrams about chemical composition. So this would be the genome project, defining the basic composition. You can also, as the last talk showed, define DNA in space, in the nucleus or in other ways, and then through time, like a cancer patient getting drugs and reacting to that at the gene level. You can follow that through time. So molecular composition through time and through space. Now every omics research has this kind of overlapping Venn diagram. Here's just the central dogma according to this 3-way Venn diagram. And there's been a lot of big science projects that have done great works to really define this and give us a strong foundation for then resting on the shoulders of that. But I want to focus now on proteomics, and that's a space that I want you to walk away with some perspective here in the next short few minutes. And one science project that was lifted up and stood up in Stockholm is the Human Protein Atlas, and it was a tissue-level map mainly focused on the spatial mapping of proteins. This was published recently. Matthias Uhlin is behind this. In Stockholm, it's about approaching a 9-figure investment to use 17,000 antibodies to track with immunohistostaining across lots of different tissues and organs. And so this is available now and starts to pull apart the multifaceted world of proteomics. So that's one piece that's really been lifted up, and private philanthropy having a key role in here from the Wallenberg Foundation. And so what other things are going on in this? And I want to focus now on chemical composition for proteins. This is an open frontier, and one that there are tools now to look at and define precisely these molecules. And so the Consortium for Top-Down Proteomics, which I helped found, is trying to put words and focus on these kinds of realities. So according to this group, you have 3 levels of analytical strategies for complex mixtures of proteins. And bottom-up proteomics, you saw it today, where even from nerve endings and sonomal cones, you have growth cones, you can do bottom-up proteomics. This is using trypsin to digest complex mixtures of proteins, and that's the dominant technology. Most labs do that. Okay? Far fewer labs are doing top-down proteomics, where we try to do direct measurement of what we call now proteoforms. This is with all the isoform information, the splicing at the protein level, with modifications, with SNPs, coding SNPs, and that is the level of the proteoform. And that I'll give you some sense of that today. And then what we've termed native proteomics, you can attack at the whole complex level. And I'll show you one slide just to put it in your mind's eye that this is possible to do top-down mass spec all the way from this third level. And the notion is that these higher levels of analytical strategies will correlate more tightly to complex phenotypes and give you more precision to uncover the underlying biology. And that's, of course, inverse from the way that the world does proteomics now. Most of it's bottom-up, and it requires more stages of inference and lots of gaps in your knowledge about proteins and lots of imprecisions, even about multi-gene families, much less isoforms, and post-translational modifications. For compositional mapping, to update you in the last introductory slide, there were 2 papers in 2014 that used bottom-up proteomics at the tissue level across lots of human tissues and body fluids, and that was 2014. So that was using bottom-up proteomics, and that's basically the state of play in modern proteomics. And so what we did is take this array of technologies, and I'll be specific about 3 of them, to implement and look at B cells in human beings. Donors, nominally healthy human beings, and so there's certainly a lot of noise in that kind of sampling, but can we use 3 different types of cell-based proteomics to map differentiation pathways and then define barcodes, protein-based barcodes, of different cell types, look at their relatedness, and the last talk gave a great introduction to this. So that's what we've been doing, is taking B cells, or all PBMCs, peripheral blood mononuclear cells, from human beings and from blood, and using a lot of flow and then mass spectrometry on the other end. So here's a dedicated flow cytometer to this project, and 3 different styles. So I'll go through them really quickly here. Part 1, Part 2, Part 3. And we're going to be doing cell surface proteomics. So there's now technologies to get 200 to 400 proteins on the surface of cells. We have 75 to 80 histone marks that we can monitor quantitatively using only 100,000 cells. And then, as I mentioned, top-down proteomics. And how will these different levels of analyses correlate to cell type? What are the rules, right? These are all undefined aspects. Output of this will be the Allen Proteoform Atlas, and I'll get into this a little bit more, but it's an open database that has the cell types in the projects, and then you can drill down on the proteins within them. So these are the 3 sources of cells. I mentioned these 2 already. Buffy coats from blood donors, bone marrow, harder to get, fewer B cells within them, and then cell lines for technology development. Underlying this project is a fair amount of hustle points on the board to develop these technologies to actually be applicable to these kinds of total B cell numbers. So here's our flow cytometer and fact system that we're very happy with. It's a 5-color, top-of-the-line system, and so here's 4 markers, and many of you are familiar with this. So we've been sorting primary cells and looking at standard B cell markers, known B cell markers, to lift up the project, and then subsorts to look at these subpopulations, and then feeding that into this 3 style of proteomics. And the first is cell surface capture. Here you can get 200 to 400 proteins on the surface of cells. It's very high fidelity. 90% or more of the proteins that you get have N-linked glycosylations and are it's been validated by many other labs, not just myself. But of course, that's high-value information of cell surface proteins, and you can use them to form bars of a barcode and then use subsequent flow when there's an antibody, if there's an antibody available. So one of the new wrinkles in the past year is how do we read these out? If you look at the cell surface, and I'm just going to use 4 cell types just as a toy because there's such little time. So here's 4 cell types. Here's all the proteins on the cell surface that we found, and you use one as bars of a barcode going this direction. Most of them are non-CD markers. So you do not have an antibody assay. So what kind of scanning technology can we use? So you can build trees and look at relationships, and that's a lot of the end stage of this project. We'll be focusing on trees, and I'll show you a couple of them in a few slides. How do you read out if you don't have antibodies? So we've thrown in this aspect to move from discovery of cell surface proteins to readout. And if there's any aficionados in the audience, it is called targeted proteomics or SRM, MRM assays. It's essentially like doing 60 Westerns in one LCMS run, even on populations of mixed cells. So and these awfully look like bars of a barcode. And so we're using this targeted technology, which is getting really close to the sensitivity of Western blots and antibodies to read out even 100,000 cells. So here's the raw data traces in LCMS. This is time. And you can clean that up, normalize it, and then it looks like a barcode. And each spike here for these 4 different cell types is an individual cell surface protein that you're monitoring. I'll call it semi-quantitatively. So that's the part 1. Part 2 is the histones. Now, epigenetics has all sorts of different meanings to different people, but the bottom line technology is we can monitor at over 36 sites of histone modification, is monoditrimethyl, and lots of acetyls. We're up to 80 different sites, and we can do this on something approaching, our goal is in a year to do on 10,000 cells. So this is our technology that works at the protein level because histones are so abundant. We can track them. And just to pick one example, here's K27, a famous repressive mark. And you can track un-mono-di-trimethyl, famous repressive marks here, the higher methylation states of K27. And clearly through differentiation, you have massive changes. You know, a 10% change on this graph is 10 million nucleosomes in the genome population, which has a total of about 100 million nucleosomes, changing. It's a massive change of the epigenetic landscape when you see changes like this. And so that's something we're doing across 80 modifications. And here we are tracking 6 cancer cell lines over here in red and 7 healthy B cells. These are front flow sorted of different types that I'm not telling you because I don't have a lot of time. But at least they segregate cleanly, except for one outlier, the cancer cell lines from the primaries from human beings. And that's reassuring. Some of you may know that the silencing marks being up in primary cells or the acetyl activating marks being up in cancer, you know, cancer cell lines are generally thought of as more euchromatic, more euchromatin in them, and that's all consistent with that kind of observation. And now we need to look carefully at the tree making based on histone, all these 80 histone marks across all these primary cell types. And piecing that apart will be the next year or so to finally get all the data and move forward. So moving to the last part, part 3, top-down proteomics. To refresh ourselves, this is this mid-level where we're not using trypsin. It's that simple. Can you do proteomics on whole proteins? Yes, you can, and it's ever more increasing in popularity and belief that you can do that measurement. So this is proteomics without trypsin, and how do proteoform level information, if you could track decorations and isoforms specifically and precisely, how does that correlate to what a cell type is, and then how cells are related to one another on a differentiation tree? So we've been able to track 850 proteoforms across 7 B cell subtypes. This is all the bio and tech replicates are at the top of the 7 B cell types. So this is a large amount of data, and 850 proteoforms tracked quantitatively, very few missing data points. That we're very proud about that. And so what's the early take on this? Well, these data are just a few months old. But some of the distinguishing proteoforms, so the proteoforms here are split this way. Some of the discerning, highly discerning proteoforms are B cell related, so that appears so good. We need to drill down more on that. CD20 is also in that group. Profilin and prothymosin, many proteoforms of these proteins carry the same information, and we need to drill down clearly more on this. But this is a new level of analysis for proteomics. And we're putting it all into a data repository, the Allen Proteoform Atlas. It's ready immediately. We have not published on this data, but that's in line with a lot of the ways in which we're moving forward as a community of scientists. And so you can drill down on these and look at the different cell types, which I blew by for time's sake, cell lines or primary cells. And then in these primary cells, okay, a CD19 pool. What proteoforms are present in CD19 B cells. These are the precise characterizations of these full-length proteins in the project. And there's about 10,000 of those currently. The goal was to get to 30,000, and we're well on our way to doing that. And importantly, you know, as this new kind of proteomics becomes possible, These proteoform identifiers. With the Consortium for Top-Down Proteomics, we've finally, after 5 years, convinced UniProt to cross-reference and be precise about whole protein information. So these proteoform numbers are durable and will be the world's information of whole, full-length proteins and all their decorations and modifications and such. And it's cross-referenced at the accession number level with UniProt. And you know, that took a while to get them to come around. And that went live this March. So I need to wrap up here very quickly. I won't reiterate what the project is about. We're trying to finish up the full 10 Bcell subtypes to complete the project's milestones. And that'll be a lot of protein level information that'll be out in just a little bit. So just the last few words before I drop the mic and get off the stage. And you know, I wanted to at least put it in the mind's eye that native proteomics or native top-down proteomics is possible. So I spent a lot of time talking about top-down. Let's go to the next level and take an entire protein complex, and the compositional analysis of that is possible to do with absolute molecular specificity. And this would be how. And we've gotten support from the Keck Foundation to do this. And we're rolling it into some of the Bcell work we're doing as well. So here's native gels. Here's a separation. There's a half a megadalton protein complex. You can weigh the whole complex by native electrospray, native mass spectrometry. So there's actually several forms of it. And then you dissociate and outpop the subunits of the complex. And you can weigh the subunits. That's the second stage of tandem mass spectrometry. And then do top-down on the subunits. And we've done this on up to 7 different subunits from the proteasome. In this case, I'm just showing you a simple case of SOD2, which you can also track the metals and other things. So this is possible with the concepts of top-down mass spectrometry. And then to put this in relation to cell-based analyses, cell-based mapping, cell-based projects. This was the inspiration of the Allen project in the lab. And I'm out of time, so I'm not going to wax poetically about cell-based science. Alan Jones and all sorts of people in the Allen operation know a lot about this for many, many years. But now it's catching on. So certainly Rick Horowitz's group and the Allen Institute for Cell Science, yeah, that's cell-based. That's important. And, but now other people are getting interested in this. There's an NIH Common Fund program, phase 2, spinning up now. The Human Cell Atlas, it's got backing. Well, Sidney Brenner's been calling for this for years, and Tom and Alan educated me a lot in the last couple days and hours about the history of this. And he's been calling for cell mapping in human beings for a long time. Well, now we have a consortium, a group of NIH, the Wellcome and Trust and the Sanger Institute, and the Broad are really pushing hard on this, and I think will launch something significant by 2018 in this space. Mostly it's single-cell genomics driven, and I'm sort of the CPO, the Chief Pestering Officer, to just lift up the value of proteomics and that we can actually make it a closed system, even though a lot of people don't believe that statement or believe the technologies are possible. But hopefully from this talk you can see that that's in the offing. And I, 4 years ago, articulated a cell-based approach to the Human Proteome Project, in which, and so you can waste 17 minutes looking at a TEDx talk or falling asleep one night. And in that, I estimated there were 4,000 cell types in human beings. We could have a lovely conversation about how I got to that number and what it's like. George Church tends to think that there's, you know, a significantly higher number of that, maybe 20,000, 25,000. But in the blood, there's about 300 cell types, and that's from unsupervised clustering of CyTOF type data. And then in each cell type, we would catalog 250,000 proteoforms, do that semi-quantitatively with top-down, some combination of top-down and bottom-up, and that would be a billion proteoforms across the human body. Includes glycans and everything, and there's a large conversation about each one of these estimates, and I'd be happy to have that conversation. So I need to finish up. Thanks to the Allen Foundation, Paul Allen, everybody here. This is I will go home reinvigorated. I have reset lots of clocks in me, and I also need to thank the Keck Foundation for that protein complex work, and it's the kinds of things that just would not be funded through NIH, and I have direct experience to get behind that. Northwestern's been great in its support, as have NIGMS, so we get a lot of support from NIH, so it's an ecosystem. And I need to thank my group. I don't have time to list all the students and postdocs. And Rebecca Gundry is also at Medical College of Wisconsin. She's the cell surface proteomics expert. So finally, I've had a fantastic 2 days, and we're not building a castle in the sky. It's got a strong molecular foundation. And thanks for the invitation. I look forward to the future. Thanks.
We have time for a quick question.
Hi. I have a question about sensitivity. So as you go up your pyramid of digested short peptides up to whole complexes, how does that work in terms of the sensitivity? How much more do you lose sensitivity? Do you require more protein? How does that work?
Yeah, all part of the technology development. There's been several billions put into making Bottom-up really work well. You could get it on a few thousand cells, get to a few thousand protein groups. You infer those peptides up to protein groups, and but you lose a lot of information as well. Bottom-up is about somewhere between 5 and 6 years behind that development curve, and the resources are highly lopsided to that top-down hasn't been given a lot of resources to develop to give a better answer. But right now...
But is there a fundamental limit?
Yeah, we need about a couple 100,000 cells to do top-down proteomics at a reasonable depth.